Thank you, Enzo. I think my favourite part about working at the MCRI is the daily reminder of the importance of the patient and the patient voice in what we do. And so I think it's appropriate then that we begin by hearing from one of these patients. So this is Tyler. He's from the patient advocacy group here in our heart failure flagship.
When I was 15, I had a heart transplant. So yeah, I was diagnosed with cardiomyopathy, but I spent probably 6 months quite ill, trying to get treatment. Since then, I've been fortunate enough to meet plenty of other people with different types of heart failure. You know, a lot of people can live relatively normal lives, like myself, and other people really have to sort of center their whole life around their condition. So it really just affects people so differently. My biggest worry, if I'm being genuine, is dying unexpectedly and just leaving that burden with my friends and family.
Unfortunately, Tyler's not alone. Around the world, 60 million plus people live with heart failure, and this is a number that's going to increase. Despite decades of advancements in this space, stubbornly, mortality remains high for those who are diagnosed with advanced heart failure. And the economic costs are astronomical as well. For those who have advanced heart failure, transplantation remains really the only option, followed by a lifelong of immunosuppression. So yes, we've made huge progress in this space, but we have to accept the fact that transplantation is not a cure for almost all of these patients. And the need for urgent therapies to target and treat heart failure remains. So in our lab, we use pluripotent stem cells to try and model heart failure and heart disease. And we believe these is a key step towards realizing the vision of developing new therapies to treat heart failure. So we work with clinical partners to take blood from patients who have inherited forms of heart disease, and then in the dish, take these blood cells, reprogram these into stem cells, and then over a 15-day period, push these stem cells towards forming two-dimensional heart cells. We can take these heart cells and then use them for a variety of approaches, one of which is to form miniaturized cardiac tissues called organoids. These can beat spontaneously in the dish, and these allow us to model disease and look for new therapies to treat patients like Tyler. We can also push these cells towards forming heart valve-like tissue for cellular repair, and also to form large sheets of engineered heart tissue patches that can be used to promote the function of the failing heart. So our cardiac organoid model is one of the most advanced in the world, and it consists of many of the cell types present within the human heart. The platform is a miniaturized platform, so these can be grown in 96 well plates, and this allows us to screen tens of thousands of compounds to look for new first-in-class drugs to treat patients like Tyler and their heart failure.
So in addition to the miniaturized cardiac organoids that are really powerful for identifying drugs and for understanding disease, we also use stem cells to make large-scale heart tissues. This is a cardiac patch, one of the projects that we have in the lab. It's made from approximately 100 million cells. And the idea of making these large clinically relevant tissues is that we can create novel cellular therapies to hopefully one day treat children with heart failure. So in this video here, you're looking at Professor Christian Brizard, who's the head of the cardiac surgery unit. He's handling one of these cardiac patches in a dish, and then he will suture it onto the outside of a sheep heart, which is what you're looking at here. So the idea being that this cardiac patch is able to act as a biological support device and increase the pumping function of the heart. One of the other projects that we have in the lab is to engineer heart valve tissue. So it's a completely different cell type, but also originates with stem cells. And the idea here is that we can remove damaged heart valve tissue and replace it with this prosthetic. And what you're looking at here in these photos on the left, this is again in collaboration with Christian Brizard and other colleagues at MCRI. He's suturing in this valve tissue in the place of a damaged leaflet. So that photo in the right, you can see the native valve tissue, and then on the left of that image, you can see the prosthetic that's been sutured in, and sort of how we envisage this being delivered as a cellular therapy. So that gives you a taste of some of the projects that we have on in the lab, but we're going to look to the future for heart disease and development.
So yeah, if we've learned today from Kevin and Holly, we already have a lot of beautiful treatments in place that allow us to help people with heart disease and also improving drug discovery. But now, if we want to look towards the future, we of course also want to come up with new approaches that help us to really understand, like, why is the heart developing how it does and how are diseases in the heart developing. So one way to do this is by combining disciplines, such as we do. We combine computational work with biology to really use state-of-the-art technology to address exactly these kind of research questions. So one approach that we can do, for example, is we can compare a healthy heart to a diseased heart. So for example, here you can see on the right side a heart that has an enlarged ventricle, which means it's weakened and there's a state of the heart that we don't want. So we want to understand, why is the heart growing like that? And one way to do that is we can use novel approaches that allow us really to look into detail into the tissue. So we really scan the whole heart and find every single gene in there and get a list of all the genes and know if they are turned on or off. And this is pretty powerful because now we can easily compare this with a healthy heart and come up with a list of potential candidates that might potentially drive the disease. Of course, only taking one cross-section isn't representative for the whole heart. So what we have to do is we need to take multiple of these sections to cover the whole heart throughout. But as you can imagine, we're generating a lot of data here. And this is really also where computational work comes in. So people like me as a software developer, we work on tools that help us to make sense of this kind of data. So I, for example, developed a tool that allows us to take this kind of data in and give biologists the possibility to really explore and mine the data. So as you can see here, this is the software, and this is one potential use case where you can see the human developing heart with exactly this kind of data. And you can look through the whole thing and give people the possibilities really to explore the whole data set at once. Using approaches like this gives us really hope to understand how the disease is developing as they do, and maybe having the chance to try to prevent them from happening in the first place. And now I want to give back to a video of Mia and Cam, who are sharing of their point of view what they think MCRI research is doing.
The research work that MCRI is doing is so important for both, like, the future of our children and all the generations to come, because it could save lives. But, like, it's really cool to be able to know that something there is, like, stem cells and grafts, like, that could actually be the cure to heart disease and heart failure one day.
Great. So thanks to all of our heart presenters for that session. I think it was really powerful hearing the patient stories in particular, and reminding ourselves that, you know, our work is far from done, and that there's really childhood heart disease has not been solved, and we have more work to do. I'm now going to